So the floor is yours, Dr. Ali.
At the onset, I'd like to thank the organizers, especially Rajiv, for inviting me to participate in this August gathering work targeting the genomics. So in relation to crop improvement. But today I will take a step what we have done over the last 6 years at IRRI, and our collaborators from CAS, jointly we did this, most of this activity. So let me start with why there is a need for breeding for resilience in the first place. The food security is a looming threat. We had seen this in 2008 when more than 70 countries were in short of food, and many of these exporting countries like Vietnam, Thailand, India, we locked our gates. And this is not very far again, but we have to produce more from even reducing resources like land, water, and the chemicals that we use. At the same time, we have to increase the quantity, and as Shafiro yesterday mentioned, that the nutritional quality is also a major concern. And this has to be produced from stable and sustainable yields using lesser inputs. In many of the rain-fed ecosystems, many people have not even seen the fertilizer bags, especially in the uplands. And the complexity of these rain-fed ecosystems is much complex, as we have seen in the past. And with this lesser inputs, can we increase higher outputs? Then coming to the disease and pest and threat that we are seeing in high input environments, as we continue to put more and more fertilizers, we are inadvertently breeding for super pest susceptible varieties, which succumb to these kind of high input environments and resulting in need for durable broad spectrum resistance. Ultimately, whatever you put on the field will end up in your plate. And therefore, we need to reduce these fertilizers and chemicals at the same time without losing the yield and quality. Coming to the recent changes in the climatic changes that we see leading to a lot of heat, increased temperatures, and flooding and drought, very rampant nowadays. The need is that we cannot breed just a single trait at a time, but think about breeding multiple traits of abiotic and biotic stress together. That's the thing. Just to highlight some of the drought stress frequency over the last 40 years, you see the red dots mostly indicating the severity of the drought. And this is the flood frequency in many parts of South and South Asia where rice is grown. But I would like to draw your attention, especially to this graph. We know the population is this green bar, but look at the oil prices are going to tip. Already it has tipped the highest point, and nowadays the oil prices are going down. But this is all going to come down as we see we are reaching the 2050 when we have to reach the 9 billion population to be fed. And look at this curve of the phosphorus. This is also tipping around the late 50s, early 50s, or you'll see the tipping end. And therefore, we need to produce varieties that will require lesser fertilizers and be use efficient. And it's certainly a race against time, and therefore the need for the green super rice concept, where we try to develop varieties, produce higher and more stable yields with lesser inputs like water, fertilizers, and pesticides. High-yielding GSR cultivars with green traits that combines many of the abiotic and disease and insect and grain quality. However, we can also reduce the cooking time if it takes just 4 minutes early cooking. That many times pots across the globe can reduce a lesser carbon footprint. And at the bottom line, it is the lesser inputs, more production, and environmentally sustainable. I would try to draw an experiment that we started in 2008 wet season, and whatever has been done is the product coming from that time. This was a variety, Huangguangzhan, which is very widely adapted and has been found in more than 10 countries across Asia and Africa. And many of this particular varieties has been identified in Punjab, and it has been nominated in ICRIP in 2 categories. And this is the genome sequence of the Huangguangzhan coming from different donors. And we integrate this with 16 donors. And if you see, many of these donors are just elite entries, not coming from drought or salinity or submergent tolerant, but just ordinary varieties, which are elite in the sense they are the best in the regions. But then we cross this with Huangguangzhan, 16 of them, and integrate this. And we started selecting for this one particular population, the first batch one, with BC1, F2, 8 of this population. We started screening for yield traits, drought screen, salinity screen, submergence, and also at the same time keeping the random set. At the same time, when we genotyped every stage, and eventually they pulled up, again is screened for a second round, and 497 integration lines, we now resequenced and phenotyped this particular thing along with the parental lines. And this has led to the phenotyping both in normal and drought and salinity conditions. And then we did the association mapping and fine mapping and design QTL pyramiding across the donors to step up and stack more genes. So this is what the experiment was. We had about 6 sites in irrigated, drought 1 site, nitrogen stress site, and cold 3 sites did this experiment. And many of these parameters we took. And coming to the genotyping part, 9 parental lines, deep sequencing with 14 to 40x, and average depth of 22x. And mostly this was done by the VGI, and 497 integration lines. We discovered 370,000 SNPs from these 8 integration line populations, having a 4,613 bins. And we did the QTL mapping using the QTL ICM software, and this is the resequencing data of the 9 parental lines. And in the background of the HSZ, we see the SNP markers. If it is very diverse, then you see many more SNP markers. If it is closely related, you see lesser ones. And by the sliding window approach, we genotype the SNPs. And this is the bin map of different populations, 8 populations that we could do with the help of VGI. And if you see, within just 2 rounds of selection, intensive selection against drought and salinity, we knocked down much of the heterozygous loci. And when you do, later on, I understood that if you do another round of such screening, it knocks down completely. And this yellow portion becomes very, very reduced, and that we experienced over time. So with this integration frequency in different lines, we try to map these traits that we are studying for cold and drought. And if you can see, this is for the grain yield and for the cold and drought. Some of these lines are beyond the parental range. I don't want you to look at this, but the bottom line is we could map many of these QTLs for yield, 66 QTLs, drought 16, for nitrogen stress 29, and 36 for cold tolerance. And these QTLs were located with the log values are here. The phenotypic variance explained is here. And much of the source of the integration came from the donors into the Huangguangzhan background, improving this. Now, coming to one of the improved lines that you can see, this is a drought tolerant check, APO, which is considered as one of the drought tolerant check, and the new integration lines that we bred out of this approach, which could withstand more than 300 kilopascals of drought stress. And these are the cold tolerant QTLs that were located on these chromosomes. And further, when we identified these integration lines, specific integration lines that were good, we which had the positive traits that were governing that. We crossed those introgression lines across the different donors with the similar Huang-Wang-Zhan background and tried to produce the F1 and F2, and again subjected them to a second round of screening for different traits. But this time we did it for 2 rounds, and that is sufficient because immediately the Huang-Wang-Zhan background is common, and immediately it gets fixed in 2 rounds of selection and achieves a high degree of homozygosity. But from the first round itself, we could develop many of the introgression lines with single traits. We had this many introgression lines developed for single traits. For double traits, we had this many. For triple traits, we had, which combined drought, salinity, and submergence. One particular line I would like to mention, this is already released in the Philippines, and which combines drought, salinity, and submergence, and it was the best in the irrigated under the MET trials of the IRRI. And this particular line is also released now. And also, if you look at the quality, people will wonder, Huang-Wang-Zhan is from China, so whether the quality will be good or not. But by the introgression frequency, that led to also changing patterns in the grain quality. It showed a very good spectrum for grain quality parameters, and that helped us to identify, out of our thousands of these introgression lines, we can now deploy customized set for any country based on their grain quality parameters using these profiles of signature profiles of the RVA pasting properties. We can match those ones with the available checks, and similar patterns can be given to those countries, especially, and we can see here the range for amylose content, because Huang-Wang-Zhan is only 14 or 15 amylose, but you can see up to 31 amylose was seen. And GT was from high to low, protein was from 7.8 to 11%. And to summarize it, what we could see is that simultaneously we can improve for multiple abiotic stress, and even if this can be extended to biotic stress. I didn't present the data for that, but this is possible, and we could recently map XA39 gene by this approach in the same population, which is already published. And the varietal QTL discovery, when it combines with omic network dissection, and ultimately this method gives you a varietal development inclusive of this whole approach. That's the fundamental thing that we are focusing. Because ultimately you discover QTLs and markers for everything. What you do with that, you have to breed varieties and give it to the farmers. And that's the bottom line. And that's the reason why this approach sounds good. And several promising introgression lines developed with multiple abiotic stress is now a reality already. And it's the Norris partners, and many of them in many countries, it has been released, and which I will share in the coming slides. And then we can upgrade this by stacking, bringing about the design QTL pyramiding, bringing 2 donors from a common recipient parent. You can cross across the 2 donors having the same background. And this can be even extended for 1 or 2 rounds to combine many more traits. And this we also did already, and we have nominated 35 entries in the advanced yield trial of the MET. And genomic-assisted recurrent selection scheme is very interesting. If you take all these introgression lines as founder lines, and you convert the HSZ into Huang-Wang-Zhan into a dominant male sterility gene is brought into that background, and this whole thing can be shuffled into this recurrent selection scheme. And again, we have done this scheme already, and I'm not presenting the data here, but we have already done this, and the results are awaited. And then pyramiding non-allelic QTLs or DNA segment, improving tolerance from different donors in the adaptable recipient parent, and resulting in superior climate smart rice varieties as per design and needs. I will just illustrate this whole thing with a set of 4 lines that has been released. 3 of them are already released. This is from the IR64 background, and you can see the kind of traits it is now having. Especially, I will ask you to focus this one: drought, salinity, submergence, and irrigated, rain-fed. It can cross even coastal saline. And it is good under Philippines, Indonesia, Vietnam, Bangladesh. It's already showing its performance. and it's released in the Philippines already, all the 4. So this thing happened in 2012 when we gave these materials to the Department of Agriculture, and they started in 2012 and 13 wet season. That's the time they can grow in the rain-fed ecosystems. In the dry season, they cannot grow, so you cannot have any crop. So the wet season, favorable season, they put these varieties, and this is the performance of this against the checks, national checks. And totally, we have now studied more than 110 locations, but 50 is presented here. And the government spent more than 20 million pesos on their own to test this. And before even the release, official release, they already started producing the certified seed of this in large quantity. One glaring example why it happened was this particular slide is very important to understand the basis. And this was in Bohol, where when the farmer is having a land like this near the coastal belt in Bohol, and he takes a nursery, and immediately seawater inundation takes place in that field after just 4 days of transplanting. And after that, you have 4 days of submergence near the active tillering stage. And they put some fertilizer after the recovery, and then the crop is at this recovery. I would just try to draw your attention to this plastic bag. It's the same spot where this photograph has been taken everywhere. So it's not a different shot. So just to keep you alert that this is the same spot, and the plant recovers, and then you enter a drought phase immediately. And after the drought, from 18th day after transplanting to flowering, and then you see at flowering a crop like this, and 1.2 tons were harvested, and that was something enormous impact in that region. And if you see the yield of the comparative yield of the other varieties that we did, we didn't get no harvest of this, no harvest. Many of the checks generally get 0, and very low in adverse condition is just 0.21. If something happens like this, just in that region is 0.21. But this particular line, GSR 8 gave 1.2 tons, and another one gave about 1.34. And the subsequent season was the more important, that was 7.23 tons of yield. And that shook the Department of Agriculture. They said, nothing doing, we don't need any release, we are just pressing the accelerator. And they started producing. This is that variety under irrigated condition. It competes very well with the hybrids and gives a good challenge for that. And then again, we did this experiment at IRRI to give the clear proof of concept to everyone. We put all the GSR lines into the pool and subjected them to the submergent tolerance test to show that how many of them, and you can see all of them are the survivors of the submergent test. And this is the IR42 dead, the susceptible check. And here you have IR Swarna sub 1, this IR64 sub 1, and this is the one I told you before, it gave 1.3 tons. And the yield was very interesting. We had some very high yielding ones, which improved upon the previous one. This is the yield of 6.78, which was comparable with the Swarna sub 1 of 7.1. But look at these ones, the new ones we identified, which even further went. But the interesting point here is the performance of other conditions is very important. Irrigated, low input, drought. Low input means completely no fertilizers were given for more than 8 or 9 consecutive seasons at that time. At this particular time, it was 10 seasons of no fertilizer ever given, of any fertilizer. And drought, and this is this. Now, if you combine these traits according to the rain-fed ecosystems that demand, say, irrigated, drought, submergence, and low input, and then take an average, then we can even weight it according to the pressure of the crop in that region. Say, we should put weightage of 40% for drought, or we can adjust that. And then design what you want to create and give to that target ecosystem, rather than giving one variety for a solution for everyone. So we can design this, and just to give you an illustration, for irrigated, we need a low input and a irrigated, and we can pick these type of entries. And likewise, for saline, we need irrigated, it should be good, saline, and drought. Because these traits never come every time. The drought doesn't come every year. The salinity stress doesn't have a high tide coming all the time. So these type of stresses especially in the eastern India, we need this kind of materials to reach. And you can see the performance of these materials under the actual, after near the harvest season, how these varieties really perform. And this was one of the very important experiments that led us. And then these are the 10 varieties that were released by the Philippines through this approach, and already released, all of them. And you can see the under irrigated, under low input, under drought, under salinity, under zinc deficiency, and the relative mean advantage over the irrigated check. So I don't want to take your time, but give you another illustration of very recently we did another scheme of starting with wheat-tolerant rice. This particular variety has been released in Bangladesh as Biridan 69, and it's a very good line, and it competes very, very strongly with the wheats in the initial part. And therefore, you can save some herbicide for saving in the lowland conditions. So we used this in a clever way. We didn't want to invest more on the lot of screening. We said, okay, we'll focus ourselves on the irrigated alone, and we'll give a middle stage, second round we'll give more stress, and the third round you'll have typically the first one. By this way, we could identify something like 2,000 introgression lines, 2,242 introgression lines. And today we have seen that many of these lines eventually made very good in the irrigated, because we were very focused on the irrigated program and to give a good answer for the irrigated. So eventually these lines now have the ability to tolerate many of the stresses. And you can imagine 2010, right? So, and in 2014, we have already nominated in the MET trials of IRRI, 20 lines we gave, 9 of them qualified for the MET 1, and 2 of them has been nominated in the national trial of the Philippines. And what is there? You can see the MET trial data. It is competing with the hybrids. All these lines were competing at the top of the top 11. And you can see the checks and their ranking in that perspective. The reason is, this particular line had, because we were very focused on the irrigated conditions, and we started selecting very carefully for this. This is one of this line. Look at this, and you can judge whether it is suitable for just competing with the hybrids. And this is another line from that particular program. To summarize this whole thing, I would say the conventional breeding versus the GSR breeding strategy, we did this job in 4 to 5 years. I showed you the evidence, and we have cut short by 50% the time, and the genetic gain is already doubled. And then comes to the genetic gain through the traits that we have incorporated into this with wider adaptation and higher grain yield and multiple abiotic and abiotic stress. Promotes lower rate of breakdown, because we are not going to dump many fertilizers, but keep them. We are trying to come at 75% level and 25% less than the site-specific nutrient management without losing the yield. So the genetic response to use efficiency is being selected, and it can be applied to any condition. So this way, we have done many PVS across Asia and Africa, but this is the line we did near to the IRRI, and we had many of these PVS, and farmers are so happy about it. One particular data I would like to share with, which was an injured data coming from the injured trial in India. We had a very good performance in the eastern India, and this is where we are trying to see the averages of this compared with the checks. You can see the checks at the bottom. And with that, we have now released almost 24 inbred varieties across Asia and Africa, 22 hybrids, and we have produced about 31,500 tons of seed, and almost 73 is nominated in the national yield trials of different countries. So in next 2 years, you can see a big flooding of these type of materials coming out, and I hope that this can produce a good impact and change the livelihoods of the rain-fed farmers and change millions of I'd like to thank the Bill and Melinda Gates Foundation for providing the funds, and the GSR project director is Zikang Lee, and David Burginson who's the Director General of ICRISAT, was our program officer in the past, from the first phase 1. In the phase 2, it was Jun Yao, and I like to thank them. They were actually very strong to support this concept, and they helped us to see this happen. And many of these partners across Asia, about 16 countries we work on, 8 countries in Africa and 8 countries in Asia. And all these GSR coordinators in different countries, we have Dr. Alexis, who is the East Africa coordinator, and we have released 2 inbreds in Rwanda and Uganda with their help, and Rosemary in Tanzania, and all these colleagues at IRRI, and the GML group, my own group, and the visiting research fellows and students, and Pauline, she is the project administrator and my secretary. Thank you very much.
Thank you, Dr. Ali, for such a nice, wonderful presentation, which has shown the light in the farmer's field. I think that's what somebody talked about, markers to market. And it is the success of this breeding approach which has resulted in release of the variety, and we hope that other countries will also follow. With this, do you have any questions, comments? Yeah. Can you just give mic behind you?
Yeah, hi. My name is Alexander Sorokin. I represent a small enterprise from France. My question will be, how much effort you are putting to identification of the genes which are responsible for these beautiful traits, what you presented here? Because there are many new technologies are coming which could accelerate your cycle of breeding, but by just stacking the traits, inducing specific modification in the genes, which would bring this specific property, what you are showing here. So is your priority just to generate the cultivar, which would be practically producing high yield, or also to identify the genes which would give you more understanding how to accelerate the breeding and how to generate even more high yield in this kind of cultivars?
If I get your question correctly, that how are we going to utilize the genes and how are we going to utilize for the genetic information that is by stacking these QTLs and genes. At the onset, I am a breeder. I am basically a plant breeder, and my objective is to reach the farmer as quickly as possible. So, as a geneticist, if you have to think, certainly these materials are a reservoir of so many traits, and I understand so far is these complex traits, there is a very huge epistatic interactions occurring between the traits. And even if you pick any few QTLs out of this, and you want to make a big improvement from that, you just forget it, because that's what I've seen. In nutrient use efficiency, I didn't show you the slides, we had to go for 5 rounds of such selections, 5, even 6, to stabilize. That itself shows that how complex that trait is. But at the same time, if you try to individually attack one QTL at one marker or one major gene at a time, it will take ages to complete the whole thing. And that's not the time that we have with us. The time is running short. And if you have to give something quickly, we have to reduce this breeding cycle quickly. And I would suggest even we should go for even dihaploid breeding or double haploid breeding in this regard. But we have certain steps by which we can reduce this. But a brute force technique that we do in the screening for drought, especially when we do drought, we are screening at something like 300 kilopascals or above. And generally 50 is considered as drought, minus 50 kilopascals is considered as drought. So we give a very huge pressure for that.
I think you can discuss with him during the,
Very nice presentation indeed. My question pertains to one of your slides in which you mentioned your breeding efforts for less cooking time cultivars. Less cooking time. You mentioned one.
Less cooking time.
Less cooking time, right. So I just want to understand whether it will have any implication with respect to the glycemic index of rice, which is already on higher side.
Actually, the cooking quality, the cooking time is governed by the GT, gelatinization temperature. And if you are able to have a variety that can cook quickly, that means it will have also softness also. Many of these varieties have softness also after cooking. And interestingly, the lines that we developed have a very low GT, and it saves about 4 to 5 minutes of cooking time. And even if it is averaged, it should be good. But coming to your question on the glycemic index, I have no direct relationship, I don't know, but there could be a possibility because basmati has low glycemic index too. So possibly there's a linkage could be there, but I don't know still. We didn't go through that.
Thank you, Dr. Ali, and please join me in thanking him for a wonderful talk.